All right. Okay, if we can all settle down, that way we can get started right at 6, so we can give our speaker the full allotted time. Welcome, everyone. Welcome, Dr. Navid. This is our second talk of the spring seminar series. So this is the last one of the spring. I'm happy you guys made it to this talk. Before I get started, I would like to welcome you all. First of all, thank you for coming. And then also I want to do a few acknowledgments and give you some history in terms of how this seminar series came about. And so first of all, I'm Adeliza Flores. I teach chemistry here, and I've been part of the organizing team for about 8 years or something like that. It has evolved a little bit over the years, and so I want to acknowledge all those people who were involved in support of the seminar series and the ones who are currently part of the organizing team. All right, so first of all, brief history. This was conceptualized by our current dean, Nan Ho, over there. She used to be a biology faculty. Now she's our dean for our division, and she was the original brains in putting this together after she was able to take part in a lot of these seminars at the lab, right? So she wanted to bring speakers like Dr. Navid over to the campus so you guys get a chance to listen to them talk about their work, right? The other people I want to acknowledge are the people who are on that side, right, in the lab. I don't think I saw Joanna. There she is. Joanna Albala is one of the people from the lab, along with Dr. Chris Culp, who have been helping over these years to bring these wonderful speakers over to the campus. Right, they look for them, they talk about them, they invite them over so we can have this opportunity. And Dr. Joanna Albala is the Education Outreach Manager in the Science and Technology Education Program at the lab. And then along with Dr. Chris Culp, who is the group leader in the Biosciences and Biotech Division. There are the two on that side who are helping us put these talks together. Right? We also, of course, want to take all the speakers who have graced our seminars here. From the LPC side, I want to thank all the faculty who have encouraged their students to attend the talk, all the staff, and also the administrators. We have, I don't see anyone besides Dan, of course, our dean, who's here. So I want to thank those people. Let me see here. There's a few more, and then I will hand it over to Dr. Navid here. Okay, so the planning team, right? I'm part of the one of the original members of the planning team, the organizing team. But I want to acknowledge Carol Schill. Where is she? She's right there. Some of you know her. She really is sort of the leader of the pack here in terms of getting all the gears running, right? All the steps in place to get us to this day. So I want to thank her. She's our STEM coordinator. I want to thank David Lee. He's here before anyone else getting all this stuff set up so that we can have nice audio and video, right? The other people I want to thank is Dean Nan Ho and her office, Linda Cross, her admin assistant. They've been very helpful in a lot of the public relations stuff and just getting the logistics going. And then the other people I want to thank from this side are the faculty members of the planning team. I think Dr. Ansel is over there, right? Mike Ansel is one of them. I want to thank Robin Rayhagen from physics. I don't think she's here today. I want to thank Barbara Zing from biology and Keith Level from engineering. I also want to acknowledge Dr. Richard Groh is here to help tonight as well. He's over there. And then lastly, right, just two deans that have helped in the past, Dr. Neil Eli, he's already retired, and Dr. Lisa Everett, our former dean. I want to thank them, or we want to thank them, because they've been instrumental in keeping this and sustaining the seminar series. And then from the lab, Dr. Vitalis and Dick Farnsworth were the two of the original team members in the planning. So now on to our seminar, finally, right? So our talk for today is on Computational Systems Biology, simulating life from microbes to humans, right? I'll let Dr. Navid give his talk, but real quickly, a few Notes about Dr. Navid. He is a computational systems biologist, so you'll learn a lot about that today, in the Biosciences and Biotechnology Division of Lawrence Livermore National Lab. He is currently the computational modeling lead of LLNL's biofuel and bioenergy-related scientific focus area, right? So got a lot of areas covered there. His research interests include analysis of complex biological problems such as host-pathogen interactions, microbial communities, drug metabolism, using state-of-the-art computational modeling. And lastly, his bachelor's is in chemistry from University of Maryland, his master's in inorganic physical chemistry from University of North Dakota, and his PhD is in theoretical physical chemistry from Indiana University, and he's also been a postdoc both at the lab and at the NIH. So without further ado, I give the stage over to Dr. Navid. Let's give him a big hand.
Thank you. I want to actually begin by thanking the organizers for giving me this opportunity to, and the pleasure of briefly giving you an overview of some of the computational system biology modeling that we do at Lawrence Livermore National Laboratory, particularly what we do in my lab. I just noticed that obviously I got the date wrong. So I guess I'm one day ahead. But anyway, so to begin with, we need to actually just examine what has happened to biology in the last 30 years. Biology used to be a reductionist science. And what I mean by that is 30 years or so ago, what they used to do is they used to study biology by just breaking up a system into its various components, whether it was a pathway, an enzyme, a protein, and just focus studying on that. And they were hoping that by examining these individual components and then getting an idea of how these interact with the other components, come up with an idea of how the whole system works, how the whole organism works. Well, as this cartoon kind of, in a, facetious way says, is, that's not how biology works. Biology is an emergentist discipline, meaning that the whole is greater than the sum of its parts. So you cannot just look at individual components and get an idea of how it works. The biology of the whole system works. In order to study a whole system, you need to examine every one of the different components all at once. Now, for a long time, we were not able to do this because we didn't have the technology. But starting in the 90s, revolution in analytical techniques were happening, what we actually call these days the omics revolution. Whenever we put an ohm at the end of something, we mean the catalog of all the components of that in a system. For example, genome means all the genes that are present in an organism. Proteome is all the proteins that are present in an organism. So once we actually had this system-level data, we now had a new problem because biology now had changed from a data-poor science into a data-rich science. And this actually was such a success that actually as early as 2010, some folks were beginning to raise alarm about the fact that it was now more expensive to store biological data than to generate biological data. That's something that biologists couldn't even dream of 30 years ago. That was insane for them to imagine something like that. Just to give you an idea of how much data is being generated, in one database, the Sequence Read Archive database that NCBI hosts, between 2012 and 2017, the amount of data that was on that database doubled 4 times. As of October of 2017, there is about 14 petabyte, that's million billion bytes of information on that database. And of course, with all of this came the realization that biologists by themselves could not analyze all this information. This was pretty much out of their league. And a realization was made that in order to use all of this data, biology needed to change. It needed to become a multidisciplinary science. The idea first was put forth by 2 gentlemen, Hiroki Kitano, who is a computer scientist at Sony, And Leroy Hood, he's one of the founders of the Institute of Systems Biology in Seattle. These guys put forth the idea that it was time for biologists to actually work with other scientists and begin adapting some of their techniques, some of their tools, in order to be able to analyze the data that was being generated. They needed to use tools that were generated by chemists, engineers, computer scientists, physicists, and mathematics. And all of these would result in a new form of biology which they labeled systems biology. Now, what is systems biology? Here you see two definitions by two eminent scientists. On the left is a definition by Professor Hans Westerhoff from University of Manchester. He says systems biology is a science that aims to understand how biological function, absent from macromolecules in isolation, arises when they are components of their system. If you're a little bit confused, it's correct definition, but you really need to sit there and think about it to realize that what he says is correct. On the right, you see a definition from Professor Bernal Paulson from University of California, San Diego, who says system biology is an integrated field of research that combines experimental and computational approaches to systematically study relationship and interaction among various components of a biological system. That one makes a lot more sense because he uses simpler words and provides more detail. And what's interesting is actually this is reflective of the kind of modeling they do. Professor Westerhoff is a big name in the field of kinetic modeling. When you do kinetic modeling, you use very complicated math, but then in order to use that complicated math, you kind of abstract the system. On the other hand, Professor Paulson is pretty much the father of flux balance analysis, one of the most popular constraint-based modeling methods. And for constraint-based modeling, you have very detailed networks and models, but you use very simple math to solve them. Constraint-based models are popular because they allow us to just get an idea of what are the capabilities of a biological system. They're not dynamic. You just get a snapshot of what a system can do. And because it's just a snapshot, one picture, you can use simple math, but then it allows you to also put in a lot of detail into your models. Kinetic models are dynamic. They actually allow you to examine the changes in a system during course of as time progresses. Now, here you use more complicated mathematics, but because these models require a lot of parameters, and sometimes these parameters are very hard to get, you kind of abstract your network. So, let's start with kinetic modeling. As I said, kinetic modeling is probably the preferred method of modeling for a lot of folks who do this because it's very useful, but the problem is that it is very, it requires a lot of information that we unfortunately don't have. However, when we do have information, they are very accurate in their predictions. And when we talk about kinetic modeling, with dynamic modeling, a lot of people think about folks sitting in some lab crunching numbers and things like that. But for me, actually, the funnest dynamic models that I usually use is actually when I play Madden. It's actually a dynamic model. It's a bunch of different players. Each one of them have, I don't know how many different parameters or how many descriptives about them. I think it's about 20 or so. Kick power, throw accuracy, tackling, things like that. And then they are grouped together in teams, and then the computer simulates it. I don't know if you guys have ever been bored enough to do this, but I've actually had the computer play the computer. And it's actually sometimes better than the game you see on the TV. It is pretty good. And it has actually a very good accuracy. They have simulated a number of Super Bowl, I think more than 10, 12, 13, something like that. And it has almost a 70% accuracy rate. In some cases, for example, the case of Patriots and Seahawks, it actually got the score right. In case of Patriots and Falcons, it actually correctly predicted that Falcons will go up and then Patriots will come back and win. So it has actually, when you have numbers, these models can be pretty predictive and pretty accurate. Now, of course, this is a game. How do we use these things to actually get something that's important? For example, can we use these models to get useful information with regards to things such as how a drug, once taken, will be metabolized, or how its time course in the body? Pretty much, can we simulate the pharmacokinetics of a drug? This is important because, you know, believe it or not, the older you get, the more drugs you take. And I'm not talking about the kind of drugs you guys think about, but I'm just talking about everybody. 81% of adults 57 or older take at least one prescription medication. Most of the time, it's, you know, it's Lipitor or something like that. Two-thirds of the seniors take 5 or more medication. That's a lot. And it's been found that if you exceed 4 or more, the chances of you kind of mixing them up together and getting some sort of adverse drug reaction is pretty high. Every year, about 200,000 senior citizens are rushed to emergency rooms because their drugs have been interacting with each other and causing some sort of adverse behavior. So here's one of those cases where we can use dynamic modeling to get an idea of how these drugs will be absorbed, how they will be distributed in the body, and how they will be metabolized. And the kind of modeling that is used to do this kind of simulation is called whole body physiology-based pharmacokinetic models. Now, again, these are dynamic models, and as I said, dynamic models require you to have a certain level of abstraction. And when it comes to the abstraction for these models, it comes primarily in the way the body is treated. The body is treated as a series of well-stirred, homogeneous compartments. What I mean by well-stirred, homogeneous is that there's an assumption made that as soon as a cell or a group of cells, a group of compounds enter a compartment, they're distributed equally everywhere. There is no diffusion gradient from one end to the other. So you have the same concentration in one end of the liver as you would have at the other end. In reality, that's not the case, but that's an abstraction that we make. And of course, everything is connected together via flow of blood. So a lot of detail is taken out in order for us to be able to actually simulate these. These models require 3 different types of data. The first 2 are pretty easy. The first one is systems data. This is just a physiological information, like, you know, what is the rate of blood flow, and what is the volume of different organs. And these things actually usually are standardized. Usually all the simulations are for a 25-year-old white guy who's 70 kilograms, and that just same set of numbers is used over and over and over again. Then there's the model structure. This is just the interconnectivity between the different compartments, and pretty much also just labeling of what is the function of the different compartments. Some compartments are just the storage area. That's usually, like, for example, fat at times. I don't know if you've ever heard, but like fat at times actually stores drug, and years later when you lose your fat, you suddenly get some weird reaction to a drug that you took 5 years ago or something like that. There are those that are for metabolism, like liver, and there are those for excretion, like kidney. The data that's pretty hard to get for these models is drug-specific data. These are things like the lipophilicity of the drug, how they're metabolized, which enzymes metabolize these drugs, what is the affinity of the enzyme for the drug, and things like that. As I said, the math that's used for these models is pretty complicated. I'm not going to go through all of them, but just to give you an idea of what are some of the important components of how the change in concentration of any compound in a compartment is measured or simulated, is pretty much one of the important parameters, or one of the important components is how much of the drug that comes in through blood is pretty much sticking into the cell. That's what this difference is. This is what goes out. This is what initially came in, so the difference is what stuck around. Then how much of the component, how much of the drug is actually being injected into the organ at any time via non-blood means, like let's say you inject something into an organ using a needle. And the last one is how much of it is excreted, either through just excretion via liver, via kidney, or for example, if it's metabolized by some enzyme. So you will have one of these for each one of the compartments that you have in the model. At times, it's also necessary to account for the delay that you would get if you take a drug using, through oral means. Here, you have to actually account for the delay in how long it would take for the drug to move from your stomach to your small intestine and then get into your GI tract, into your blood. And at this point, you also need to account for how much of the drug might just be excreted through the colon, how much of it might be metabolized by the bacteria in your stomach, and things like that. So these are additional formulations that you need to add to your model in order to get an accurate prediction. Now, a few years ago, we were interested in actually doing a drug-drug interaction analysis for 3 popular drugs. One of the drugs that we were interested in is called theophylline, which is a bronchodilator. Now, this drug is not being used that much in the industrialized world because it has a very low efficacy, but it still is being used for some cases, in cases, for example, if a patient is resistant to steroid-based drugs. And it's also being used a lot in the third world because the budget for drugs in these countries is pretty low, so they find the cheapest drug that might help their citizens. So actually in the third world, theophylline is used as one of the most popular bronchodilators. One of the reasons that theophylline is not used in the West is because it has a very narrow therapeutic range. It's only 10 to 20 milligrams per liter for blood concentration. So as you can see, this narrow range means that any small overdose could easily get you above that 20 milligram per liter range, and you could have some sort of adverse drug reaction. And that's why you get a high frequency of side effects. There's also a high instance of toxicity in elderly and the young. The elderly primarily because the amount of enzymes in their liver is usually lower than healthy young folks, and it's also that the excretion through kidney is also lower than most young patients. And in the young, it's because just their metabolism in the liver is not matured enough. The second drug we were interested in is called ciprofloxacin, which is a wide-spectrum antibiotic. The reason we were interested in it is this is one of the main drugs that's used when you're treating anthrax. But the reason that's also interesting with regards to theophylline is because it treats a variety of different infections, one of them being for respiratory infections. Now, ciprofloxacin by itself actually is known to have a variety of different adverse drug reactions. One of the weirdest ones, which I have no idea why it does that, is apparently if you take it, there's a good chance you will rupture your Achilles tendon. Actually, this did happen to my brother. So, and the third drug that we were interested in including in our model is the most popular drug in the world. Anybody knows what the most popular drug in the world is? Caffeine. It's caffeine. I don't know what you... All right. Caffeine actually is very complicated when it comes to theophylline, primarily because actually in the body, when caffeine is metabolized, it produces theophylline. The enzyme cytochrome P450 isosome IA2, 1A2 demethylates caffeine and produces theophylline and 2 other byproducts. The same enzyme then demethylates theophylline and gives you 3 other byproducts. Cyte1A2 also metabolizes ciprofloxacin. So if you are familiar with your enzyme kinetics, you know that right away you have a problem because now you have 3 compounds fighting for the same enzyme, so you have competitive inhibition right there and then. The thing that makes caffeine actually more interesting is because caffeine not only is competing with theophylline for Cyte1A2, it's also producing it's also producing theophylline as it's being demethylated. So anytime you take caffeine and theophylline, immediately you all immediately get more theophylline than you usually would get if you take just theophylline by itself. So the first thing that you do when you develop these models is you formulate the model, formulate the mathematics, parameterize them, and then you run a simulation and see if your model agrees with experimental observations. And here you see the result of our simulations for the 3 different drugs. Now, one of the things you want to do is you want to make sure you use at least 3 or 4 different enzyme, 3 or 4 drug concentrations, because you don't want to have your model be some sort of like artifact of just, agreeing with one concentration. So it's important to make sure that you can simulate the behavior for a variety of different concentrations. Now, when we were studying theophylline, one of the things that jumped out was that when we were reading about the instances of adverse drug reactions, we noticed that the majority of the cases were for women. And one of the unfortunate byproducts of living in a paternalistic society is when we simulate things, when we measure things, we do everything for men. As I said, a lot of these models just automatically dump in numbers for a 70 kilogram white male into their model, and then whatever the outcome is assumed to be for everybody in the entire human population. But if you look at actually the physiology of men and women, you see there is a big difference. Women, on average, are 20% smaller than men. The blood flow through different compartments is different. Women have more fat on their body than men. Men have more muscle, which makes it problematic. For example, if you have a drug which is lipophobic, then this drug will just circulate in the blood a lot more in women than it would in men. So, given this, we decided that when we were developing, we developed actually 2 separate models, one for men and one for women, with the data you see here, and then we decided to simulate to see what we get. Now, one of the things that we were examining is that when folks have done clinical studies, they have found that same group of people, let's say the 25-year-old 70 kilogram man, one of them will achieve therapeutic dose after only 300 milligrams of theophylline. The other one might need 1,600 milligrams. There is a large variety, and this is primarily due to differences in the liver. It doesn't matter what gender you are or things like that. And given this large variety, doctors have come up with a way of trying and avoid adverse drug reactions. They have developed what they call a dosing, initiation, and titration protocol. And how this works is, for patients, adults, 16 to 60, and any children that's heavier than 99 pounds, given today's kids, all of them are almost 99 pounds, so I guess everybody almost. They start out by giving the patients 300 milligrams a day of theophylline, let it go for 3 days. If the patients don't exhibit any kind of adverse side effects, then they up the dose to 400 milligrams a day. And at this point, they stop. They don't increase the levels anymore for elderly over 60 years old. But for patients who are younger than 60, if they don't exhibit any kind of adverse drug reactions, they up the dose to 600 milligrams after another 3 days. And so here you see the result of our model simulating this. And one of the interesting things that you see early on is actually, given the differences in metabolism and clearance for the elderly, and how we simulated the elderly, by the way, was we read a bunch of papers and found out that the average metabolic concentration of the enzymes for elderly compared to a young person is about 2/3 the value. So that's what you see here. So they pretty much, their liver metabolizes the drug about 66% of the rate that a healthy young person would do. And also their clearance through the kidney is about about 30% of what it is for healthy adults. So when you combine these things, you see actually it is a very, although this thing is based on trial and error, it is a very good protocol because the amount of drug that both elderly and the young are getting is about the same. Now, as I said, there is a wide range of different levels of metabolism among folks. So we wanted to know, all right, for an average man and average woman, how much reduction in the activity of the enzymes in the liver is allowable before they start feeling the effects of adverse drug reaction, given the same, treatment, if you give them the same DIT. And one of the things we found out was that for men, a person can have as low as 65% of the activity of other people, and still they will not observe any kind of adverse drug reaction. That's what this is. For women, anything less than 85% activity, they will start actually beginning to feel it. So given this, one of the things we were then thinking about is like, okay, is it possible for us to use our modeling to come up with some equations or some method for doctors to be able to predict actually a personalized dose for the patients? So what we came up with is, we came up with this way where the doctor will give a patient a 5 milligram per kilogram of their weight dose of theophylline, and after 2 hours, they collect a blood sample and measure the concentration of theophylline in the blood. That would be this parameter here, C star max. And then doing a whole number of different simulations and linear regression, we came up with a number of different equations for men and women, which allow us to, just using that one parameter, predict how much the metabolism of the drug in this patient's liver is different from other folks. And also it allows us to actually come up with a dose that they can use, which would not cause any kind of adverse drug reaction. And here you see a simulation. And for 3 different patients with very different levels of enzyme activity in their livers, you see that all of them are pretty much getting the same level of the drug, very far off from the even it's not even close to the level of where they might observe any kind of adverse drug reactions. So this is actually one very useful way of using the models, is to actually come up with new helpful mathematics to help drug treatment. Next, we wanted to test to see, okay, can we examine how drug-drug interaction differs between men and women when they take these different drugs together? Here you see that when men take 600 milligrams of theophylline, 750 milligrams of ciprofloxacin, and 133 milligrams of caffeine, all of them together, they still do not get anywhere near to the level of where they might have any kind of adverse drug reaction. On the other hand, women, if they take 1/3 of the amount of ciprofloxacin that the men took, caffeine and theophylline, they already are up there. So this just shows that it is, when it comes to at least theophylline, it is really about time that we begin to, do 2 separate measurements and 2 separate dosing for men and women, because there is a big difference when it comes to how men and women metabolize drugs. Finally, we just decided, why not? Let's see what is the final frontier when it comes to examining differences between people. Race. So we already knew that women were more susceptible to drug-drug interactions than, adverse drug reactions than men. So we simulated a group of different groups of elderly women taking the 400 milligrams of ciprofloxacin. And one of the things that we found out is we used these numbers which were reported in a paper by Carpenter, which just pretty much gives you the different body weight and height of women in a variety of different races. And one thing that jumped out at us was how Asian women, taking the same dose that other women you will be able to tolerate, could very easily start displaying adverse drug reactions. Now, so far I've been talking about using these models for pretty much pharmacokinetics, but we use these to simulate the interactions on a variety of different kind of systems. One of the projects that I'm involved in is just, we are interested in generating biofuels. One of the missions of the lab is energy security of the country. And although maybe currently bioenergy is not exactly the top of the list of DOE, but things come and go, and eventually bioenergy will be one of the renewable sources of energy that we will be all using. So one of the projects that we are involved in is we are interested in how we can use the biomass that's produced by algae. These are organisms that just use carbon dioxide and light energy to produce biomass. How we can use this to produce biodiesel. And one of the things we found is for one of our algae, when we co-culture it with a bacteria, we find that actually the growth of the algae improves. So we did some studies, and we have a hypothesis that the actual compound that causes this behavior is actually a popular phytohormone called indole 3 acetate. And the next thing we studied is, okay, generally, how much of this phytohormone do bacteria produce? And one of the things we found out is actually they produce very small amounts of it. So if they're producing very small amounts of it, the only way that this will work is if they are, within close proximity of the algae. Otherwise, the concentration just will never be large enough. So one of the simulations we did is we took 12 of these bacteria and kind of clumped them on the surface of the algae. And what we found is that, yep, if you do that, you would get a local concentration somewhere right underneath these, about 52 nanomolar. The threshold for actually observing the improved growth is 50 nanomolars. So here we can see that it is possible with a few of these cells directly attached to the surface to get the desired behavior. Here we simulated if we disperse the bacteria all over the surface, and we found that, okay, no, that actually the concentration on the surface drastically dropped. Here, we simulated, all right, let's just move the whole clump a little bit further off. Does it need to be actually touching the surface of the algae? And again, the concentration drops. So based on this, we get an idea that, okay, the only way that you can get the observed behavior for this small number of cells is if they're actually directly attached and they're clumped. So, dynamic models give you the ability to do a variety of different kinds of examinations. And if you, as I said, if you ask anybody who does modeling between constraint-based modeling and dynamic modeling, which one you prefer, they all will say dynamic modeling because it is much more informative. But the problem is that, as you saw, these are very parameter-intensive models. And given all of our advances, we are generating a lot of information. The one thing we're not generating is kinetic parameters that will allow us to get an idea of quantitatively what is the interaction between these different components of a system. And that's where constraint-based modeling comes in. Constraint-based modeling is very popular and is used a lot because what it asks of us is exactly what we have, and what it doesn't require is what we don't have. What it asks of us is an annotated genome. An annotated genome provides us with a parts list of an organism. It pretty much tells us all the enzymes that an organism can have at different times in the cell. And once we have that, we then have an idea of what reactions they can catalyze. We can go to a database like KEGG or Biopsych, and once we have all of these different reactions, we can then combine them together and get a reconstruction of the metabolic network of an organism. Now, sometimes when I'm trying to describe this to my friends, at this point they kind of like, so now what? And to be honest with you, at this point I really think about, usually I think about what the reconstruction gives you is something like the highway map of the United States. By itself, it really doesn't tell you much. You need to have a question, an objective, and you need to have actually constraints that you put to get some sort of information out of it. So for example, let's say you want an objective. All right, I want to drive as fast as possible to Livermore, California. All right, there's your objective. Okay, where do you want to start? Let's say New York City. There's one constraint. That reduces the number of different paths you can take. All right, what else? I don't want to go through any of the red states. That takes a whole bunch of stuff out of there, right? So, and then like you can say, I want to go through highways and things like that, and this diminishes the number of different routes you can take. Although in the end, believe it or not, even if you do that, you will get maybe 50 or 60 different ways that you can go do the same thing. This is called the generative solutions. And the same thing happens with our modeling. So when we're developing these constraint-based models, the first thing we do is we reconstruct the network, and then we formulate it in a mathematical formalism in a what we call a stoichiometric matrix. The stoichiometric matrix, each one of the rows is one of the metabolites that's in the network, and each one of the columns represents one of the reactions that affect the different metabolites. As I said earlier, each one of these requires an objective. The objective for constraint-based modeling that's most often examined is production of biomass, growth. Now, other objectives can also be examined. For example, you can examine production of a compound of interest, let's say a drug or biofuel. But 99.9% of the constraint-based modeling efforts examine maximizing growth of an organism. And for that, you need to have an idea of what is the biomass composition of the organism. And how that is done is we just write one reaction that's added to the rest of the reaction network, where we just break down the different components of a biomass, such as DNA, lipids, and proteins, and then we break it down even further until we get to the level of metabolites, such as cysteine, uridine, and some fatty acids, and then we'll just have a coefficient next to all of these, and what we do is then we optimize this reaction, the flux, which is the reaction rate through this reaction, given the constraints that we put on the system. Now, the method that's most popular when we're doing constraint-based modeling is called flux balance analysis. These models are usually constrained with experimental measurements, if we have any. We also make sure that the models agree with fundamental physicochemical laws. By that, I mean we make sure that they obey laws of thermodynamics. We make sure that they're not unlimited import of nutrients, unlimited export of waste materials. And then we make one final assumption. We assume that the system's at steady state. What that means is we assume that the change in concentration of the metabolites inside of the cell is 0. During the whole time, the concentration does not change at all. Once we put all of these constraints, then we can just use linear programming, which is very close to linear algebra, to maximize, to come up with a feasible flux pattern. Here, I emphasize feasible because it will be one of the various solutions that you might get. We will get a feasible flux pattern result in the optimum value of our objective, which was growth. Now, flux balance analysis is very popular, and there is a large toolbox of different kinds of analysis that you can do with flux balance analysis. You can examine the robustness of the system to various kinds of environmental perturbations, genetic perturbations. You can examine the range of different fluxes that can flow through each one of the reactions. You can even do some crude pseudo-dynamic simulation where you can get an idea of how metabolism of cells affects the concentration of nutrients in the surrounding media. So, it's one of those things that in my group we use a lot. And during my last 11, 12 years at Livermore, I have developed 5 or 6 of these. The one that is one of my favorites is the first one I developed, which was for Yersinia pestis, which is the causative agent of bubonic plague. At that time, this was the first genome scale model of a CDC category A pathogen. And the reason we are interested in it is because of its biosecurity implications. Bubonic plague is a very nasty disease. Actually, it's not a human disease. It's a disease of rodents. But at times, the flea jumps off of a rodent, bites a human, and then the human gets bubonic plague. Now, if the disease remains bubonic, then the only way that the disease can be transferred from that human to another is if another flea bites the human and bites another human. But at times, the disease actually mutates and becomes pneumonic. That means at that time, the disease can actually be transmitted from person to person pretty much by coughing. And when that happens, then it gets really scary because the rate of transmission drastically increases. Now, a lot of times when people think of bubonic plague, they think of, Rutger Hauer and throwing corpses over the walls and things like that, things from medieval ages and, sword and sandals movies. But it's kind of hard to actually really fathom what a nasty disease this is. By all accounts, during the course of history, it has killed over 200 million people. To just give you an idea how much that is, that's more that was killed by plague alone than have been killed by all of the wars pretty much all the wars that we fought since the beginning of time. And another thing that a lot of times people think about is like, well, again, it's an old, from medieval times. But even today, there are still outbreaks and pandemics that occur There was one a couple of years ago in Libya. There was another one that occurred actually in Madagascar. And what was scary about that one was they actually were finding strains that were antibiotic resistant. And that was really freaking people out, because then we really will be in trouble. And even in the United States, each year there's a handful of different cases that pop up, mostly in Southwest, a lot of them actually in New Mexico. We actually had a colleague who used to work in Los Alamos with pestis, and he had a saying that in New Mexico they say, New Mexico, home of the flea, land of the plague. So, so one of the things that is most interesting about the Yersinia pestis is by law, we cannot study them in a lab. We cannot do any kind of genetic mutation. That's where actually modeling is useful, because we can do whatever we want in a model. So that's exactly what we did. We did a whole series of what we call in silico gene knockouts. We knocked out the genes one by one to see, okay, if I knock this gene out, will the cell survive? We did this for different kinds of environment. We found some genes that were always essential. We found some that were essential when they were in a rich medium. We found some that were essential when they were in a poor medium, some that were under anaerobic conditions. So all of these actually can serve as a drug target. Now, another thing we could do is we could do what are called synthetic lethal mutations. These are the cases where if you knock out one gene by itself, you don't get any kind of, you don't observe any change in the behavior of the organism. But if you knock two of them out, then you see a phenotype change. And this is interesting because this allows us to actually think about some sort of combinatorial drug treatment. So these are the kind of things, that analyses that we can do with FBA with regards to treatment or identification of drug targets. But as I said, it's always problematic because we do get a variety of we do get these degenerate solutions. And it would be a lot easier if we actually had an idea of which one of these pathways to really focus on. And as I said earlier, the way to do this is by adding more and more information to your model. And at Livermore, we've been working on developing a method which we call GXFBA, which allows us to actually incorporate gene expression data, differences in gene expression between two different situations in our models to get an idea of how metabolism changed when you went from condition A to condition B. The result you see here is when we simulated metabolism in Yersinia pestis after it was treated with an antibiotic. And here you see those pathways which were upregulated, the big fat red ones here. Obviously, these are important for the organism. Those that were downregulated, the blue ones, and those which were not changed at all. So once we have this additional information, we can actually say, all right, I got 80 different targets now, but which one of them actually is upregulated? Which one of them is important when I treat this organism with some sort of medicine? So as far as FBA is concerned, so far I've been talking about mostly simulation of single-cell organism. However, the next frontier in computational system biology is modeling of cellular communities. This is because we're now getting more and more information about, omics of ourselves, our ancestors, our pets, our food, and important microbial communities such as communities in the ocean or our microbiome in our gut. We want to be able to simulate the behavior of these organisms, especially when we change the environments. Unfortunately, we cannot use FBA for this kind of simulation because FBA will predict at times biologically infeasible scenarios. Let me give you an example. Here you see a co-culture of two organisms, A and B. Each one of them is growing happily. Organism A produces something that organism B likes. Organism B produces something that organism A likes. And organism B produces a product that we like. So if we just ask FBA, all right, give me a scenario where I can get the most amount of product out of this system, and we don't give it any additional constraints, FBA will say, well, I have the perfect solution. A should not grow anymore. It should only produce that compound that B likes, and B should not use any of this newfound nutritional wealth. It should just produce what you want. Mathematically, this is the perfect solution. Unfortunately, biologically, this is unfeasible. Because what we're doing here is we're ignoring the actual, the objectives of the cell that is actually hardwired in it. So if we want to actually really engineer these systems, we need to account for the differences and the trade-offs between the different objectives of the cells and what we want to get out of the system. And to do that, we need to do what we call multi-objective flux analysis. Here we actually measure quantitatively how change in one objective affects another objective. And what I mean by objective here, again, is a biological task, production of something, production of ATP, combating some, some disease or something like that, some biological task. And the result of these multi-objective analyses is what is called a Pareto front. A Pareto front is a situation where if you increase the value of one objective, it will result in diminishing the value of another. For example, here, if you increase the value of urea secretion, you will be reducing the value of ATP synthesis. That's a Pareto front. Now, I'm not going into mathematics of how we do this, but one of the things that I want to bring to your attention is each one of the points you see on the Pareto front is one simulation of a linear program to solve. Now, if you want to do something like 2, 3 objectives, 4 objectives, depending on how many points on the Pareto front you want to map, you can do this on a laptop. It's not a problem. But if you want to increase the complexity, and that's what we want to do, we want to be able to actually do real-life computation, microbial communities, then the problem becomes taxing, computationally taxing. And that's what my group has been working in, particularly working with Dr. Mark Grissom, my postdoc, who's here. We've been developing this tool which actually allows us to do up to 10 objectives on a regular laptop within a day, which is remarkable. I mean, this is the highest that anybody's done for biological organisms. And here you can see the results that you could get of this kind of analysis. Each one of these tiny lines is actually one possible phenotypic scenario. And if you map the result of experimental measurements into this space, you actually get an idea of what is the actual objectives of the system. It might not be maximizing growth. It might be, okay, I want to conserve carbon as much as possible, or I want to produce maximum amount of ATP under these conditions. So this allows you to get an idea of what the cell wants, and then you can kind of massage it in order to get the most out of what you want. Like, for example, here, one of the things we analyzed was production of hydrogen, and one of the things we found out is there's no way you can produce hydrogen and not reduce the growth of the cells. So unless you come up with some other way, you cannot get extra hydrogen out of these cells. Now, up to 10, beautiful, but we want to really do much more than that, and that's where Livermore comes in. In order to do this thing, you need to actually, if you want to expand beyond 10, you want to do 20, 30, then you need to actually be using high-performance computing, and that's where Mark is developing this serial version of the code, which allows us to actually run it on Livermore high-performance computing and do analysis for a large variety of different objectives. And in the end, we're hoping to be able to do this to examine natural as well as artificial communities and assess the trade-offs between different system tasks and traits, and then be able to kind of see how much leeway each one of these traits have and how we can engineer the environment and engineer the system in order to get the most out of what we want to get out of the system. So in summary, as I said, there are two types of modeling, and the one that usually is the most informative is the kinetic models, because they give you an idea of how the system evolves. And when data is available, these are pretty accurate models. Unfortunately, there is not that much data available, and at times, either we need to make really, really drastic abstractions of the system, or we just need to switch to something else, which is constraint-based modeling. These are powerful tools which allow us to examine the capabilities of an organism, and we are constantly developing new methods to improve the capability of these constraint-based models with regards to multicellular systems. Eventually, we want to do all of this in order to be able to design new experiments, identify new drug targets, and pretty much this is going to be linked directly into synthetic biology. As a matter of fact, recently we developed a new group in our division, which is just system biology and synthetic biology group. And the more information we get and improve the models, the better the predictions will be, and eventually expansion of these networks would help us get better with predicting. And these are all evolutionary processes, so any new information comes, you can just continually improve the models. And in the end, I would like to acknowledge my collaborators. There's a whole bunch of them, some folks from LLNL and folks from a number of different universities, and our funding, which is mostly from Department of Energy and Department of Defense. And I want to thank you for your attention and glad to answer any questions you have.
Now is the question and answer section of the talk. I'm sure you guys have a lot of questions. There's a lot of information there. Who wants to go first? Anyone? Here's one.
Hi. So when you were talking about the kinetic equations for metabolism, those are some pretty complicated equations. How do you go about deriving those?
They are just basic enzyme kinetic equations. It's just because they become more complicated the more compounds you add, right? It's, the problem becomes when you actually have to have 150 of these things, all of them coupled together, and then something goes crazy. Then you really need to be digging, going through all of these, and usually what you find at the end is somewhere instead of a 1, you put an L, that sort of thing. So otherwise, these days there are all sorts of programs that allow you to solve these coupled ordinary differential equations, whether it's MATLAB or Mathematica, which are very useful. In terms of complexity, a class in enzyme kinetics, and if you ace it, you will be able to easily do this, no problem.
Any other questions? Anyone else? I have a question. You have a question? The existing question. Can you tell us a little bit about how you got to where you are now? Essentially, where you started, what got you interested, all those nice, wonderful stories.
As you heard, I started out as a chemist, and it wasn't easy because I'm colorblind, and I kept flunking the the titration part. I remember that I had this Austrian professor who was just really a stickler for accuracy, and he would take off 1 letter grade for 1% error. So if you were 3% off, you're already almost at D. So that was one of the reasons I decided, all right, maybe I'm not an analytical chemist, because that was what my interest was. I was really good with my hands, and I wanted to do things like chromatography and things like that. But then I changed my mind. I decided, okay, I'll do inorganic chemistry. So I went to North Dakota, started doing inorganic chemistry, and I really didn't like it. And one time I almost blew up a lab because I somehow had a leak in one of my vacuum lines and was producing liquid oxygen, which actually is pretty explosive. So then I decided, all right, this is not for me either. But one of the things that I was always good at was physical chemistry, mathematics, and things like that. So when I went to Indiana, I started working with Professor Peter Ortoleva, who is a physical chemist. He was doing, at that time, mostly modeling of geological basins, pretty much production of oil and things like that. Well, I wasn't very interested in that. So he said, what are you interested in? I said, well, I saw this map of metabolism outside of one of the doors in the hallway. I would like to do that. And he said, fine, go ahead to do that. And that was it, love at first sight. It's just it's one of those things that you need to find what you're good at and what you're interested in. One of the things that I found out quickly is I kind of get ordinary differential equations. That's might be actually one of the things that I might have been selling dynamic models too much, is because I like them. I see an ordinary differential equation and kind of in my head already see how the system's going to behave. That's the kind of thing that interested me, So that's how I got to it. And one other thing that I give, I would say is stick to it. Believe it or not, I've had professors who told me, forget about graduate school. Let's talk about how you're going to get some internship afterwards. I've had, professors who mocked me because of, my immigrant background. Things like that. Grow a thick skin and stick to it. Believe it or not, most of them don't know what they're talking about. You know what you want to do and do it.
Hi, I have a question. Does your system allow to like model new growth of like brain cells in the brain given like stimulation, like a drug?
Which model we're talking about?
Any model that can model.
I mean, you can
like,
abstract, okay. One of the things we're doing, one of the projects I'm working on now is we are interested in examining the life course of immune cells, for example. Are they generated, during the course of your life, or you get a certain amount of them in the beginning or right after a disease and they stay that way? That's the kind of thing. There, I wouldn't do any kind of genome scale modeling. That'd be insane. There, I will develop some sort of a abstracted dynamic model, and yes, then you can model those. It's one of those cases where it just depends on if you are somehow interested, if one of your cells is a mutant and is going to behave differently. I mean, for neural cells, mostly that's not going to be a problem, right? Unless it's a cancer cell, of course, and then you got other problems there. But actually, cancer is one of the most interesting things to study. And actually, if you think about it, FBA is cancer. That's one of the things that one of my advisors first told me when I went to the lab. It's like, no cell maximizes growth. The only one that does that is cancer cells.
Hello. You said that you used like more than 10 components into, like the high level modeling. That's when you showed like
Yes.
it had all the different colors. How could you possibly read that? Because all the other graphs is like you could see, like the legends and all that.
Right.
And how could you possibly read that?
Okay.
And what does it mean?
I'll tell you the big secret of scientific publication. Unfortunately, we always have to have a pretty graph. I mean, I don't know if you've seen, there are these graphs that people put in their papers. Looks like a bunch of ants attacking a single thing. Nobody can get anything out of that. But people put it in there to say, look, the complexity of my work. When it comes to this, what I do is I take the Excel sheet and I read the Excel sheet. This doesn't tell me anything. This is for the person who's reading the paper. So they can get an idea of, wow, he did 50,000 different phenotypes. And then they go to the supplementary. If they're really, really interested, they need to go read the supplementary material. Yeah, you're right. I mean, a lot of these things, you need to read the, just to give you an idea, Mark today was like working on something and he's been working on it for 2 weeks, running the same model over and over and over again, and he cannot redo the result that the original paper was reporting. So eventually he went to the corresponding author's website and found out that, oh, they forgot to mention that you need to incorporate these different compounds in the model in order for it to be running. So, I mean, a lot of times I say if you're reading something, make sure you read the supplementary materials, and sometimes even beyond supplementary material. If there is a website, go to the website and check to see if there's anything that they didn't include in the original paper.
Any other questions?
My question is, why are we still using antibiotics? Can your model, help us come up with something else other than antibiotics? Because with all this resistance and
That's the goal. The problem is that whatever we come up with, the cells come up with some way of fighting it. One of the things that a lot of people, for example, are now working on is phage therapy, which I think is probably the most, one of the most promising. This was something that initially was being used before antibiotics. They were actually treating a lot of bacteria and pathogens with phages to kill them. But then antibiotics came and they're like, why do we need this? This stuff works great. But, you're absolutely right. The way we're using antibiotics, whether it's to feed the chickens or whatever, it's crazy. I mean, soon all antibiotics will be useless. And when that happens, then we're in deep, deep trouble. So I think a lot of people right now are focusing on alternate paths of killing bacteria, making it actually, and another thing is trying to get them to be not broad spectrum. being targeted. Because one of the things that we are finding more and more is when you take these broad spectrum and demolish everything in your gut, you actually are helping some of these antibiotic resistant bacteria that were being controlled by your original gut microbiome. Now that all the competition is gone, they're like, yay, and they go crazy. So, I mean, a lot of times actually you get diseases that you didn't have after you get an antibiotic treatment. So I totally agree with you. At this point, there are certain things that are being worked on, but you know, they're all in early phases.
Okay.
Any other questions for Dr. Navid?
How can bioengineers use your data?
Bioengineers use my data?
Yeah.
Well, it depends on what they want to do, though, right? If they, for example, want to come up with a way of engineering a system, they could use something like MOFA's data to say, all right, it seems like under different conditions, the cell is willing, for example, to forego growth for something else. So then they could do some sort of analysis to see, all right, is it possible for us to make it, instead of doing objective X, which it was doing under condition Y, if it allows us to do the objective we're interested in under the same conditions. I mean, a lot of this stuff that happens and we get when it comes to engineering is because we're not accounting for the regulatory mechanism that's overlying the metabolism. I mean, a lot of times we're just focusing on one level, but biology is multi-layered. And unfortunately, we miss with one and the others either fix it back to the way it was or go somewhere where we really don't want to go. So that's the thing. They need to do this to kind of get an idea of, get environment-specific results. That's one of the problems. I mean, and I think the only way we can get really is if we can get some ways that we will have stable environments. Like, again, in the whole bioenergy study, one of the things one of the reasons we're doing the whole analysis is because these large algae farms in Texas or whatever, they're crashing out of nowhere. Today they're fine, the next day they're crashing just because, let's say, some storm blew some bacteria off the sky and dumped it in there. So, you know, the only way they can get this not to do that is if they kind of can make the environment stable. But how do you do that for a football field size raceway, right? So that's the thing. I mean, they can get some ideas of what the cell is willing to give. The rest of it is up to them and coming up with ways of doing it.
Way in the back.
So my question is kind of two parts. One, what do you see as the future of this field or this line of research? And two, what's the biggest roadblock do you see getting there?
Okay, the roadblock is the easier work. The roadblock is right now we do not have any kind of quantitative interaction data. This is something that I've been to a number of different conferences, and Bernard Paulson, the guy I showed earlier on, he's just constantly advocating for all of us who do this to actually, you know, get together and send letters to NIH to kind of start a process. I mean, these kind of numbers were being generated in the 60s and 70s. But then suddenly this whole new technology in genetics came up and everybody gave up on it, right? So we kind of need to go back to doing that sort of thing. That's the roadblock. As far as what's the next step, a lot of it for the dynamic simulations, the problem is a lot of these systems are multi-scale, and when you have multi-scale, particularly with regards to time, things at some point begin to crawl because you have one process which is happening at a microsecond time scale and you have another one which is happening on a weekly scale. The code will run and simulate the behavior for the micro scale. So you will not be able to see any kind of change in that, you know, for the weekly scale. So one of the things we need to come up with is ways of actually solving both at the same time. And there are colleagues at the lab who are doing that. They have come up with a variety of different ways of actually solving two tracks and then kind of at some point catching up and trying to reconcile the behavior. The reason that you cannot do it usually is because what might happen in this column, the fast column, might affect the behavior of this one. So if you run them, decouple them, you might actually miss some behavior. But these guys are actually doing, developing new methods to do that. As far as constraint-based modeling, the next step is trying to actually, what's one of the things that actually we are thinking of doing is actually accounting for interactions with regards to, physical interaction between the cells. That's not what's there. Right now, these cells are just producing biomass, which is a blob, which is kind of like, even when you're doing kind of like two-dimensional diffusion models of them, they're just, the biomass just flows between different compartments. There is no shape to it. Things like that are the next step that we're interested in doing.
Thank you.
Yeah.
Any other questions? Well, if there, oh, I see one up.
I have a question. What was, in all your research, what was the biggest surprise, or are you like, whoa, I didn't realize, or didn't think that would happen?
Oh. Biggest surprise. Well, I'll tell you which was the biggest surprise and kind of a little bit of. We were working on this one organism. I was telling some of the students about them. We were interested in trying to improve the consumption of a specific compound, pyruvate, by an organism. And I was pretty sure that we would be able to just come up, perhaps we would be able to express some additional transporters and we'll be able to get this organism to consume more of this compound, and then everything would work because we had these engineered co-cultures which would produce biohydrogen. But I started to doing multi-objective flux analysis and mapped the experimental result in it, and the first thing that popped out was this organism is not carbon limited. This organism is actually light limited. So you could feed it all the carbon in the world. It's not going to grow any faster. The only way you will get it to grow faster is if you can somehow make it produce more of these light centers on its surface, which is a possibility, but I think at some point you'll get with the problem which the cell has this limited surface area. So that was one of those things where like, okay. I don't think we can work with this thing.
And with that, I think, let's go, I'll thank Dr. Navid for a wonderful talk. And we hope to see you guys back here in the fall for our next series.